All right. So reminders. One, please remember to fill out the doodle poll before class on Friday if you have opinions about the review session. Right now, when I last looked, there was like a four-way tie between different times. So, um,
I'll be in Lycoming. I don't even think it's 4 hours away. A game.
well,
Anytime on Sunday.
well, like I said, if you have not responded, you should respond. Um, Also, remember that you have the adaptive immunity assignment lab due tomorrow in lab. A bunch of you have already emailed it to me, but if you have not, make sure I have it tomorrow in lab. And know that the vaccines problem set is due on Friday, by the end of the day, Friday. All that stuff is posted. Tomorrow in lab, we're going to do the lab practical. So make sure that you come with your lab notebook, as it will be an open notebook practical. And also, please bring your laptop or some other device tomorrow, because I'm going to give you some time during lab to do your evals. Because me fitting in information, it's already going to be hard enough for me to talk about vaccines in one day on Friday. I don't have time for evals on that same day. Yes, Brian.
Labs need to be like attached to the notebook.
Um, Technically, yes. I would like it to be like you definitely are not. I don't want you using electronic notebook. You know, it needs to be like a paper notebook. Um, I so not a lab I worked in. Lab one of my friends worked in. Um, boss who used to come around and do stuff like this. And I'm gonna tell you what he did. I'm also gonna tell you I once said this right before the practical. I was like, this is actually like a test someone I knew used to do in their lab with lab notebooks, and everyone freaked out and was really worried that I was gonna do it, and like I wasn't. But just know that like technically in a real lab, one of my friend's bosses used to walk around the lab, pick up their notebooks and shake them, and then when like the random like paper towel that you put in to like write notes about something wasn't actually attached and fall out, you'd have like half your lab notebook on the floor, and he'd be like, yeah, that's why you're supposed to attach things and actually write it in your notebook. And then he'd like walk away. So no, I'm not going to take your notebooks and shake them. um Technically in a lab setting they should be attached. I'm not going to check it super closely. So that's my answer. um so yeah. I think those are my reminders. Are there any other things that you guys have questions about or that I should be reminding you about? Yes, Juliana.
I don't know if you can exactly answer this, but
Uh-huh.
for the climate,
Uh-huh.
would you say, I know it's a cumulative exam, but would you say it's more heavily on with the newer information?
Um, I would say it is weighted towards the newer information. As you might notice, as you start looking at a lot of the newer information, it does like tie in really closely with the older information. So it's very hard for me to like give you an exact percent because sometimes I'll look at a question and be like, is that new or is that old? Um, and I would say to use the old exams as a guide on that. Other questions? All right. So today we're going to talk about cancer immunology. Um, I'm going to do a really brief set of a couple key points about sort of cancer and cancer biology, just to make sure we're on the same page for some things we're going to move into in cancer immunology. I will also tell you that I think that I teach cancer immunology a bit differently than some other immunologists tend to think about it, which you will see as we get a little bit into this, which I actually think is I think some of the stuff that I'm going to tell you about cancer immunology that doesn't necessarily show up in your textbook is really important stuff to think about. So just be aware of that. But again, before we can kind of think about sort of the cancer immunology stuff, I want to just point out a few things about cancer biology in general. And so there are a couple of terms that we need to think about. Two of those terms are here, which are transformation and mutation. So cells can undergo mutations, which are changes in their DNA following exposure to mutagens, or just because of errors made during replication. And one thing that I want you to remember is just because there is a change in DNA, that doesn't mean the protein's going to change. That also doesn't mean that even if the amino acid in a protein changes, that doesn't mean that like the protein is going to totally get changed. And if the protein does get changed, that change could be beneficial, that change could be neutral, or that change could be bad. And so I think that sometimes people have this idea in their head of all mutation is like catastrophically terrible. And so know that there are different kinds of mutations that have different kinds of impacts. Yes, there are some mutations that can lead to a lot of issues, but also know that there are some mutations that do things like, at least in one of my ancestors, gave that ancestor blue eyes. That isn't, a catastrophic, terrible thing. And so know that different kinds of mutations can have different kinds of impacts. If a cell that's in culture gets certain specific changes, that cell can become transformed, and be known as a transformed cell. And transformed cells differ from normal cells in that they have some kind of issue with their normal growth and death processes. So normally a cell undergoes normal growth and normal death, and so we sort of are at homeostasis with the number of new cells we have being born, the number of old cells we have that we're getting rid of. If we either mess up how frequently a cell divides, or we mess up how frequently cells die, we can end up with excess numbers of cells. And so here we're thinking about a specific type of mutation that will impact either a growth or a death process. And if you have cells in a dish that get their growth or death processes messed up by mutation, we refer to those as transformed cells. That transformation can happen as a result of various mutations. It can also happen because of viral infections and some of the DNA changes that may happen with viral infections. But as you can see, note that every mutation doesn't necessarily transform a cell. Every viral infection doesn't transform a cell. It has to be a mutation or a viral infection that does something to hit growth and death processes. If it's a viral infection or a mutation that like makes your eyes blue, that's not normal growth or death processes, so that's not going to transform cells. And so that's one thing that we can sort of talk about first. Sometimes people talk about transformed cells and think about kind of transformed cells as cancer in a dish. I don't love that idea, because realistically, for something to actually be a cancer, we need something beyond transformation. So just transformation isn't actually enough for a cancer. We actually need this process called oncogenesis to happen to actually get to cancer. And so to actually get cancer, to actually have a tumor, we need oncogenesis to happen. Transformation is really something that happens in a dish of cells. Oncogenesis is where you actually have that cell dividing too much and causing a problem in an organism, in an animal. And so you can see oncogenesis is what's actually happening in an organism, not in a dish. And what you can see here, and what you'll see as we talk throughout, is that we usually need some additional kinds of genetic changes for a cell in order for that cell to be oncogenic. So we need to mess with growth and death processes in the cell, but we usually also need to impact some other processes in addition to the growth and death processes. Very classically, people talk about these things called the 6 hallmarks of cancer, or 6 things that are needed for oncogenesis. If you take Cancer Bio with Dr. Dunaway, I believe the cancer biology field is actually at 12 right now as the actual number of things they need, not 6. But these 6, which are your textbook shows, are sort of the classic ones. I'm going to mention one other thing as we go. But the idea I want you to see is that a cell needs to actually be able to do a lot of things to actually really be a cancer in an organism, like stimulate its own growth, ignore growth inhibiting signals, avoid apoptosis, develop a blood supply, be able to move around the body, and be able to replicate constantly. So there's a lot of things that that cell is going to need to be able to do. To make that cell go from a happy, healthy cell to be transformed into a tumor cell. Huh? Giantmicrobes.com. And one other sort of general piece of cancer biology that I want to mention is something called the multi-hit hypothesis. And again, we're going to think about some aspects of the multi-hit hypothesis a bunch of times. In the first slide, I told you about transformation and issues where a cell gets sort of a problem with its growth or death processes. Might not surprise you to learn that growth and death processes are kind of important for the cell. And so they're usually under multiple layers of control. It's not like we have one thing keeping the cell in check for growth or death. We usually have like multiple backups because we really don't want to mess up growth or death processes. And so in general, for a cell to be transformed, and definitely for a cell to be oncogenic, the cell doesn't need one change. The cell needs multiple changes, referred to as multiple hits. So we might need to see multiple mutations happening in a cell before we actually get to a cancer. And so you can see at the top, maybe we'll have a cell that makes one mutation in making a progeny cell. So you can see, it's got one progeny cell that has a slight change. It's a little pinker than the other one. And maybe that cell now can divide a little faster. Maybe while that cell is dividing a little faster, it makes another mistake. And so it makes another progeny that has additional mutation. Maybe now that it's made 2 mistakes, we've gotten rid of 2 of the controls, and so now it's easier for that cell to mutate again. Now perhaps it's easier for that cell to mutate again. And whenever we actually look at any particular tumor, we see that that tumor has had to undergo multiple hits, both to really destroy all of the backup systems for growth and death processes, and to get to some of those other hallmarks of cancer. And so this process takes a long period of time. It's not like one day you had happy, healthy cells, and the next day you had this tumor. There's this sort of time process where there's these intervening cells that are picking up more mutations over time. And each individual tumor in each individual person will be sort of a completely different cell that has gone through a different sort of journey or set of hits in order to get there. And so no tumor is exactly like any other tumor. Yes, there are some proteins and some genes that are commonly impacted, but basically what you can see here is if 3 different cells lead to tumors, they're going to go through 3 sort of totally different paths. So each tumor is going to be unique. and have picked up kind of a different set of hits. And again, those are things that are going to become important as we move forward. So I've already kind of implied this in terms of what types of things might impact cancers and lead to some of those genetic changes that I mentioned previously. There are a number of different sort of environmental or lifestyle factors that may lead to different kinds of mutation. You know, we can think about smoking as a very famous example and lung cancer, but there are lots and lots and lots of sort of environmental things that may lead to different kinds of cancers. There are also a number of different viruses that lead to or are associated with different types of cancers. And one thing that you might notice is, you know, there are some viruses that are on this list of viruses that can cause cancer that are viruses that are found pretty commonly in the human population. Epstein-Barr virus, or EBV, being particularly important. EBV is the cause of mono. It's estimated that by age 30, like 90-something% of people have been infected with that virus, but it doesn't mean that everyone's going to get Burkitt's lymphoma, because in fact people need this plus some additional hits to make that happen. So note that these are some of the things that are related to causing different types of tumors. So this is a spot where I kind of am going to divert a little bit from all of the things that you might see in a traditional immunology textbook about cancer immunology. And this is because of, I guess, my age, or when I was sort of training in graduate school. Because if you look at an immunology textbook right now, you look at your immunology textbook, you will see the cancer immunology chapter. And it will be this lovely chapter. And in fact, if you talk to people in a lot of biotechnology, pharma industries, and medicine, like cancer immunology is like a huge, hot field. When I was in graduate school, some of my professors, one in particular that I'm thinking of as I talk about this, said that cancer immunology didn't exist and that there was no such field as cancer immunology. And it was ridiculous for immunologists to spend any time talking about cancer. And so I want to actually talk a little bit about, and that was the view in the field for a while. Now, if you look at your textbook, you'll be like, the textbook does not mention anyone ever thought this was controversial. The textbook only says that like this is a big deal. And they will never say why anyone thought that this wasn't a big deal. And so I want to tell you a little bit about kind of some of that argument, because I think that thinking about some of these details actually informs a little bit of how we think about immunology and how we think about cancer immunology, big picture. And so, as I said, this one professor would get really kind of outspoken in some of our classes and be like, yeah, he's like, I don't know if I believe should be this whole field of cancer immunology. I don't know if I believe that cancer immunology is a thing. And he would say, I don't believe, or he did not believe that your immune system did anything about tumors, did anything about cancer. And so he might say, well, yeah, okay, fine. We know that some viruses cause cancers. And so, like, yes, we can all understand why immunologists care about that and how we can, like, think about immune responses to these viruses and stuff. But, like, the lung cancer that's coming from somebody smoking, some cancer that's coming from some kind of genetic predisposition, some cancer that's coming from one of these lifestyle factors, that has nothing to do with your immune system, is, again, what this professor would say. And he would say, you know, if you have some random other kind of cancer, if I get skin cancer from being out in the sun too much, my immune system is just not involved. Again, is what this professor would say. Why might you think that? Or why might you sort of think about that idea of your immune system not doing anything against the non-viral cancers? The viral cancers, kind of obvious. But the non-viral stuff, all the other reasons why cancers might exist, why might you say, yeah, there's no role for the immune system here? Yeah, Shelby. Okay, so you're on the right track. Yes, so we're not talking about pathogens. What were you going to say, Jonah?
It's all self cells.
So self cells. Yeah, yeah. It's all self cells. These are self cells that we're trying to say your immune system is making a response to. And we've spent all this time this semester talking about how we don't respond to self cells. And now we want to have a whole chapter about our response to self cells, because these are self cells that have sort of gone wrong. And so he's like, this doesn't make any sense. Your immune system isn't doing something about these self cells. Like, no. He was just like, no, this is just not a thing. And like I said, there were people in the field that really thought that way and felt really strongly about the idea that, yeah, immunologists can concern ourselves with viral cancers, but the non-viral stuff is just not like part of our field, not part of our wheelhouse. But there were also some pieces of data that started to kind of argue against that. One of those pieces of data came from looking at tumor tissues. And so here you can actually see two different tumor tissues, one a breast cancer, one a skin cancer. And in both cases, when you look at the tumor, this is also true when you look at a variety of other tumors, and the tumor cells are pointed to helpfully by the red arrow for those of us who are not great at histology, myself included. Whenever you look at a tumor from a patient, you find that tumor is full of lymphocytes. There are lymphocytes all around the tumor. So any tumor you look at has a ton of lymphocytes that seem to be around it, involved in it, hanging out there. And so that made some people say, well, if there's a whole bunch of lymphocytes suddenly there, it kind of seems like maybe your immune system's trying to do something. If all the lymphocytes are trafficking to that site, like, what's up with that? That's weird. And a couple of other pieces of evidence sort of started to, again, really push people into this idea of this field of cancer immunology. One of those things was the huge number of AIDS patients, people with HIV, that we started to see as time went on. These patients are immunodeficient, so their immune system is getting destroyed. They're losing part of their immune system because of HIV infection. That's what the disease AIDS is. And so what happens to people with AIDS is that they will have all sorts of infections that lead to disease that most people would be able to fight off easily. And so we can look at a whole list of different kinds of infections that cause disease in AIDS patients that are sort of reasons why those patients die. But it turns out AIDS patients also get more cancer than other patients. And so when we look at the list of all the different things that are problems in AIDS patients, they get cancers. And some of them are cancers that are viral cancers, but some of them are non-viral cancers. And so that made it look like, so if you're missing an immune system, you get extra cancer. So that kind of makes it sound like your immune system's doing something about cancer. Similarly, investigators have developed a number of different types of mice that are immunodeficient. And so these mice are missing largely their adaptive immune responses. We either have nude mice who don't have a thymus, and so they have no T cells. Of course, then their B cells also have problems with activation. We have skid mice who are missing the common gamma chain receptor, and thus can't respond to a ton of cytokines, make no B cells or T cells, mostly no NK cells. They got a lot of problems. And what people were realizing is that, people have been using these mice in experiments for a lot of years. I don't know how many years, but a lot. That's great. Usually you get some nude mice, you do your experiment for a couple weeks and you're done. If you got some of these mice, some skid mice or some nude mice, and you just didn't do an experiment with them, you just let them live in your mouse facility for a year and didn't touch them, didn't do anything to them. It turns out in most cases, those mice die of nasty cancers. And most people hadn't been letting their mice just sit around for a really long time before. But if you just actually took these mice that were missing an immune system and just let them sit around, didn't infect them with anything, didn't just sort of let them hang out, they all got all sorts of crazy cancers. And so again, immunologists were like, it's like the immune system does something about cancer. Because if you don't have these adaptive responses, you're suddenly getting too many cancers. But I want you to keep in mind this problem of antigen that we mentioned before but it's a self-antigen. What's up with that? And we're going to see that sort of theme come up over and over again. And so there were a number of observations and experiments done that really kind of laid the groundwork for this field of cancer immunology. One such experiment is shown here. And so in this experiment, we can take a mouse and we can give that mouse tumor cells from another mouse. But those tumor cells aren't live reproducing tumor cells. They're tumor cells that have been killed by radiation. So you can see we give this mouse irradiated tumor cells, so dead tumor cells. Okay. If we were to have given the mouse just the tumor cells, then in fact the mouse would have gotten a tumor and the mouse would have died of the tumor. But if you previously give the mouse irradiated tumor cells and then give the mouse actual viable tumor cells, the mouse is protected and doesn't get the tumor because, in fact, the mouse has somehow made an immune response that protects it from tumors. And so, again, you can start to see this immune response. And if you actually do something like put in or give an irradiated tumor and then give the mouse a different kind of tumor. So again, you gave it a irradiated red tumor and now you are going to give that mouse viable blue tumor. The tumor actually grows. So this in fact is a specific response. It shows all the hallmarks of being part of an adaptive immune response. And experiments like these, again, furthered this idea of sort of cancer immunology. I guess one other thing I will say, and again, we can get to this later on, is in those arguments about whether or not the immune system does anything about cancer, whether there is a field of cancer immunology, I think that one other argument, like one big argument, is it's a self-cell. Like, how are you suddenly going to have these heroes coming in and getting rid of these bad self cells? But the other thing that people were kind of thinking about is like, if your immune system does something about cancer, then like, why do so many people end up with cancer? Like if the immune system is actually good at this, then why, like, in the world does it happen as often as it does? And so that's something else that we can again think a little bit about. When investigators tried to further understand what's going on with immune responses to tumors, we found that much of tumor immunology was mediated by T cells. And so there are definitely roles for B cells and antibodies, but T cells really seem to be the big players here. And so here you can see a mouse with a tumor. That mouse got that tumor because of some chemical that was put on the mouse's skin. So this is like not a viral tumor. We basically put some chemicals on the mouse's skin, ended up giving that mouse tumor cells. And we can take some tumor cells from that mouse. If we, again, put the tumor into a similar mouse, the tumor grows. It's like an active, bad tumor. This mouse is probably going to die. But if we somehow cure this mouse of its tumor, and then we sort of give it the tumor back, so we got rid of the tumor. Here it says it as resecting, but however you want to get rid of it, and then you try to transplant back the same tumor, now the tumor doesn't grow because the mouse made an immune system or an immune response. But like, it's the same mouse, so that's a little bit hard to understand. Like, it has an immune response here, but it doesn't have an immune response here. Like, what's up? That broke people's brains for a while. Or if you take T cells from this original mouse, you give them to a recipient before you give that recipient the tumor, the T cells can actually kill the tumor. So again, oh my gosh, the T cells can do things against tumors. That's awesome. That's really exciting. That's one thing that immunologists were like, how do we deal with this? How do we make this work? But the other thing that, again, broke people's brains for a long time was, well, why, if the T cells work so well in this mouse, why didn't they work in the first one? And why didn't they keep the first one safe? And so that's another thing that we're going to sort of have to think a little bit about through this process.
So the one that the eradication of the tumor, that's a different mouse? They gave the T cells from the original tumor mouse?
Yeah. Yeah, these are a different mouse. One of the questions that sort of people started thinking about was the antigen question that I've already introduced to you. Sort of this question of, what the heck? These are self cells. Are there antigens in tumor cells that could allow the immune system to differentiate those tumor cells from normal, healthy self cells? And the answer is yes, we have found antigens that are on tumor cells that are not on normal, healthy self cells that we can have, that T cells can differentiate. In the case of viral tumors, it's kind of like obvious. They're antigens from the virus. So that one's not really like shocking to any of us. But we can also think of some other types of antigens that we see in tumors. There are different ways we can define those antigens. This slide actually is dividing them up in two different ways. So you can see that there are kind of three different things shown here, but you can also see there's something listed as TSA and TAA. And so when we think about tumor antigens, we can think about two big classes. One class is called TSA, which stands for tumor-specific antigen. So we have some antigens that are tumor-specific antigens. Alternatively, we have TAAs, which are tumor-associated antigens. I mentioned sort of the viral protein. I guess a viral protein would be like would sort of be a TSA, though we don't often talk about it quite in that way. And And these three examples are sort of famous examples of how we might get some of these tumor antigens. The TSA that is shown here is perhaps one of the easiest ones of this whole process to understand. If you think about it, in a cell that has undergone all the mutations to have oncogenesis, it had to undergo mutations. I already said that in the beginning of the sentence even. Like, the point is that it had mutations. Those are changes in its DNA, and that leads to changes in its proteins. There are different amino acids in the proteins in that cell than in other cells. And so the self-peptides of that mutated cell are different than the self-peptides of all of the normal cells. And so that mutated gene leads to a new protein that now gets presented on class 1. That protein wasn't in the thymus because it only is existing in this cell that has undergone this mutation. And so that would be sort of this altered self-peptide that would be a tumor-specific antigen. And one thing that is sort of important about if you think about that tumor-specific antigen, it is going to be really unique to that cell and that person's tumor, because it's the specific weird mutation that happened in that person's tumor. It's not like you're always going to find like that same tumor-specific antigen in every single person who has a particular type of cancer, because every single person probably had to pick up some different mutations. And so this one is going to be very specific to just that tumor and not a lot of other tumors. Alternatively, one thing that we will sometimes see in a tumor cell is that sometimes tumor cells will change the amount of a particular protein that they are translating. Perhaps they will start transcribing too much of some gene, translating too much of some protein. That might just be because we've messed up regulation in that cell. That might be because the cell needs a lot of that to be a tumor. As we're making more of that protein in the cell, that is going to result in more of that protein being presented on MHC by that cell. And so we might just have so much more of the antigen that now we're actually having a higher probability of T cell activation because of overexpression of that normal protein, too much of that normal protein. The third alternative part of this is that in some cases, well, in a bunch of cases, we know this is a thing, there are genes that are in your genome right now that you are not at all transcribing at all. You're not translating. And that's because you don't need them right now. They were like genes that helped you be a fetus. You're not a fetus anymore, so you don't need to make fetus proteins, right? You turned those off. You stopped making them a long time ago. Sometimes when a cell is oncogenic cell, a cell has sort of started picking up mutations for transformation, its regulation of gene expression is messed up. And so that cell starts just making genes it's not supposed to. Like, it'll start making embryo genes and fetus genes when it's not an embryo or fetus, because it's just not good at doing regulation anymore. And so sometimes we'll start to see inappropriate expression of genes that are normally only expressed in the embryo. So these are going to be kind of new antigens. You can see some data on this here. So the table on the left is showing you the names of some different types of antigens that are found in tumors. It's mostly showing you viral antigens for the TSAs, because it can't be like amino acid number 53 got changed in Mike. Like, it's not going to list every individual person's TSA up here. It's trying to like be a little broader. And then here are some examples of TAAs, either things that are getting overexpressed or things that are inappropriate for a differentiation state. And just another example, there are a couple of things called alpha fetoprotein, it's only made in fetuses, fetoprotein, and carcinoembryonic antigen, only made in embryos. And you can see in most patients, we don't really have those being made. You can see in some of the, we usually say really high numbers of alpha fetoprotein mean that it's being turned on. We have different types of non-cancerous diseases. We don't really see that turned on very much. If we actually look at types of cancers, we start to see those embryonic antigens are actually turned on in certain types of cancers. And so you can see that we're starting to see we see these embryonic antigens being turned on in cancer cells quite a bit. There's one section of this lecture that I cut out when I was thinking about stuff for time purposes. I'm now feeling sad about having cut it out. So I'm just going to say like a sentence or two, even though it doesn't have its own specific slides. Don't get super hung up on it unless you want to like come have some discussion for years in my office. One other question that some people might have is, well, what about signal 2 with some of these antigens? So yeah, okay, fine, we're presenting self-antigen, but how are we actually activating the T cell with signal 2? Again, I could talk about this for a very long time, but what I will say is that is related to some things that we now know about the role of the innate immune system in cancer development. And so the innate immune system is definitely getting activated inappropriately here. The innate immune system is also sort of doing some stuff wrong here to make this happen. I just left all that out today because it sort of went down a whole other track I didn't feel like dealing with. But one example is that we know that certain issues with DNA damage, DNA repair, DNA replication, things like that, all trigger this one set of PRRs. And so when you're starting to get some issues with DNA, you may be triggering PRRs and getting signal 2 or excess inflammation. And that's related to some stuff I work on in my lab. So again, I could talk about that for about 4 years, but I'm not going to. And so in starting to understand all of this and all of these observations and realizing that there is all of this information about the immune system and cancer, we've realized that there is an additional hallmark in addition to the 6 that I told you about. So originally, cancer biologists talked about there are 6 hallmarks of cancer, but immunologists said, no, guys, we actually have a 7th one we have to add. You guys and so the 7th one has been added. Like I said, in the field, they actually are like at 12 right now. But we're just going to talk about the 7th one that the immunologists added, which is that what we have realized is that to see an actual tumor in an organism, to get to the point where you have a tumor that is like causing problems in your body and it is enough of something that's going to get like diagnosed, that tumor had to figure out a way to get around the immune system. And so in fact, one of the properties of oncogenic cells is ability to avoid the immune system. And so in fact, the immune system's pretty good at dealing with tumor cells. And tumors that are actually causing an issue in an organism have figured out a way to get around the immune system. And so, we can think about the fact that, yeah, this mouse that has a tumor, its immune system is not doing anything about that tumor by the end. You okay? Okay. its immune system is not doing anything about tumor at the end. This tumor is evading this mouse's immune system. But if we do other things, the immune system can kill it, like use other cells, go in another mouse. It's just that somehow that tumor became evasive in the original mouse. And so the idea is that all tumors seem to have to get this ability to evade the immune response. And this has led to a really important theory that describes kind of what we actually think the relationship is between the immune system and cancer. And so I'm going to show you the steps of that theory on the next 3 slides. It has 3 steps and there's 3 slides. Oh my gosh. So you want to kind of pay attention to that. And then I'm going to kind of come back to this kind of slide and think a little bit about it big picture. Okay. So what we think actually happens in an organism is that at first we have some cells that get mutations that maybe start on this path to oncogenesis, but that your immune system, largely involving T cells and NK cells, will recognize and eliminate them. And so you can see here's some healthy tissue at the top. We have some cells that go wrong, that get some of these weird mutations. And then the immune system comes along and actually eliminates those cells, and we get our tissue back to normal. And so this is sort of this phase where there's elimination. And so now what we might imagine is that in every organism, there are always cells that are making a mistake and going down this path of oncogenesis. Like every day, the organism has a like microtumor, or like one cell big tumor, right? Some little bitty tumor that's like starting. And most of the time, your immune system just comes in and is like, nah, and wipes it out and gets you back to healthy tissue. And so you're frequently seeing situations where you're picking up mutations and then the immune system is eliminating those cells. And so maybe that's happening. And you could imagine, again, this happening in organisms like every day, that you're just cleaning up these like microtumors. They're 1 cell big, or they're 3 cells big in this image. They're not something that's actually going to like cause physiological changes. They're not things that your doctor is going to be able to diagnose. Your doctor is not going to find one rogue cell in your body. They're going to just get eliminated. That's going to be it. But as time goes on, you might have additional cells picking up some additional mutations, and eventually, because of sort of unfortunate natural selection, some of those cells are going to pick up mutations that allow them to start to avoid immune responses. And so eventually, you're going to get to this second phase where the tumor and the immune system are sort of at equilibrium. You're getting a few tumor cells and they're actually sticking around, but they're not like increasing out of control. So now we don't get rid of all of the cells that were problematic. We maybe leave a few around, that we don't get rid of every last one. There are a few left, but they're not like going out of control. You're at equilibrium. And so this is known as sort of this equilibrium phase. And this might happen because the cells now aren't just having 1 mutation. Maybe these cells have 2 mutations. Maybe we have a original cell. I'm not very tall. Maybe we have an original cell. It picks up a mutation and actually no one cares about this cell. But this cell's a little more able to mutate. Maybe it picks up 3rd mutation and this cell gets killed. But this cell still has, because this has like mutation A and mutation B. we still have some mutation A cells around. Eventually, this cell might pick up not A and B, but A and C. It might sort of keep trying with mutations. And now, maybe that mutation, that combo of A and C, allows us to evade immune responses, while A and B allowed us to sort of allow that cell to get killed. And so eventually we're going to end up at this phase of sort of equilibrium, where the cancer cells are actually persisting, or at least sort of the pre-oncogenic, the cells are sort of persisting, they're around, but they're not increasing dramatically in size. They're probably not causing any physiologic problems. There's probably still not enough of them that you're actually going to diagnose.
Is there a reason they're more resistant, or are they just slipping through cracks?
They have randomly picked up a mutation that allows them to survive. So it's sort of a natural selection thing. They didn't try to get that mutation, but if they happen to get a mutation that allows them to survive a little bit better, then they can slip through the cracks. Does that make sense?
I didn't know if it was a specific mutation that's more resistant than
the cell is not like trying to like, please give me this mutation. I want it. It's just picking up random mutations. If it gets something that allows it to evade a little bit, it will. And then eventually we get additional mutations, and now we get mutations where we're going to get full-on escape of the tumor cell. And so now the tumor cell is going to be able to divide without stop. The immune response is not able to stop this response anymore. Before, maybe you have a cell that's dividing a little bit excessively, but you're sort of keeping it in check. Now you got no check. The immune system is totally avoided. We are now at the point where, so here's, a cell that had one mutation that we could kill. Here's a cell that had another mutation that we're sort of in equilibrium with. Now we pick up some more mutations, and that cell is just no longer controllable. And that cell now will get to the point of fully escaping. And so you can see, we're going to have some variants over time, and eventually there's going to be this one variant that just completely avoids immune responses. And now you're going to get that cell to divide so much that cell is going to actually be measurable, that cell is actually going to cause physiological problems, and that we're actually going to see disease. And so part of the idea here is that in a patient who actually shows up at the doctor's office, they've been at stage 3. They're at stage 3. They've probably been at stage 3 for a while. These are not like the cancer stages that you talk about when you're diagnosing cancer. This is sort of this immunoediting theory set of information. And so, if somebody shows up at the doctor's office, their tumor already knows how to avoid the immune system. Because if you could diagnose it, it already has gone through this, and it's probably been avoiding the immune system for quite a long time. And so, again, you can see sort of that multi-hit hypothesis here. And also, I want you to just notice, so this shows you the diameter of a tumor and how many times a tumor has to double. I actually find this part more interesting. So notice that for a tumor to be visible on an X-ray, that tumor needs to have, I really can't read, does that say 10 to the 8? I can't read that. Yeah? Yeah, okay. Okay, hold on. I think that says like 10 to the 8. I don't know. Well, this is 10 to the 12th. This is like 10 to the 9th?
Yeah.
Okay. All right. So 10 to the 8 cells. So that means that in order to be able to actually see a tumor and diagnose it, it has to be 10 to the 8 cells, which is 100 million cells. Think about how many divisions that cell had to undergo before it got to 100 million cells. And that's the first time we're diagnosing that tumor. So this has been going on for a long time to get to 100 million cells. And we had a lot of time earlier to pick up mutations and to sort of learn how to avoid the immune system. You might first feel that tumor at 10 to the 9th or 1 billion cells. That patient might die at 10 to the 12th. which is 1 trillion cells. So note how much doubling this takes. This also you could imagine, explains some of the reasoning why we see cancers in older individuals, because it takes time for any mutated cell to go through the processes of picking up enough mutations to escape an immune response and to actually get around this whole process. And so you need this sort of goes into this idea of really needing a whole lot of hits. And it starts to tell us how important this evading the immune response is in actual thinking about cancer in a patient or in an animal. Any cancers that, again, we actually diagnose are all cancers that are immunoevasive cancers. So let's see here. Shelby, you had a question.
Is this whole process fine for people who have previously had cancer more likely to get symptoms again because you just have the most recent cells?
Yeah, generally if you are, you're killing like these cells down here, but you might not be killing these cells up here. And so eventually you can lead to that happening. Also, and so again, you'll get new mutations and sort of a new tumor. The other issue is that we can't always get rid of all of the cells, even down here. So sometimes yes, there were 10 billion, or there were 1 billion here. We did surgery, and we got rid of 999,990,000, but we left 10. So that's the other reason why people may end up with that cancer coming back, because we may not actually be able to even get all of these cells. And if we did get all of these cells, we probably didn't get all of those up there. Depends a lot on cancer types. Other questions? I felt like I was seeing hands coming up. Yeah, Juliana.
I don't know. When you were talking about like the mouse experiment with injecting the T cells and everything, I was wondering. Sorry, I'm trying to figure out how to say this, but like you said, if you inject the T cells, it will eradicate the cancer. So then, for like this part, like when the tumor evades that, are they evading those T cells in particular?
Yeah, so the next few slides are going to be about how some of the evasion works. And so we're going to specifically address that. Other questions? And then I have another point I wanted to make. That was basically the same. So I'm saying this now because I don't know if I'll actually get to this point going forward. But one of the things that you want to think about with all of this is, if we think back to that mouse experiment, you could be like, well, look, that we know that we can make the immune system do something to cancer, right? The problem is that when we see a cancer patient, the cancer patient already has a tumor that's evasive. It already has gone through the steps that we're seeing here. If I had a crystal ball and I could say this is the exact tumor that's going to happen in each person, I could vaccinate them all 20 years before. But I don't have a crystal ball to know what tumor you need to be vaccinated, what mutations you are going to pick up in 20 years to vaccinate you against them now. I only can find that out after you've had all those mutations, which is also when you're being immune evasive. So yeah, there are a bunch of things we can do in mice when we know what tumor we're going to give them to show that we can make the immune system not have that tumor. In the cases of viral tumors, we just vaccinate against the virus. Yay, HPV vaccine, which actually, if you look, there's a data that came out last week that actually showed how many deaths have been prevented at this point by the HPV vaccine. The HPV vaccine is a spectacular vaccine. And so, like, if we know in advance, then yeah, we can use this information really easily. The problem is we don't often know in advance in the way we do in a mouse experiment. But I need to tell you what the evasion mechanisms are. Because there are a few different evasion mechanisms. So one of them is that as our tumor cells pick up mutations, one of the mutations that we frequently see is that our tumor cells will stop making MHC class 1 or will have less MHC class 1 on their surface. And so this you can see might be sort of the original tumor cell. It's got what, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 MHCs on its surface. It's probably going to get killed pretty quickly. But it might pick up some mutations, so it has less MHC on its surface. And as time goes on, we might end up with a cell that has very little, but now it only has 2 MHCs on its surface. So that antigen is getting presented less. And so we're evading the immune system because we're presenting antigen less. So that's one way that we tend to see tumor cells evading immune responses. And so here you actually can see MHC staining, and you can see some places in the tumor basically are like MHC free because we have just downregulated class 1 expression. In addition, I've told you that cells that are under stress will start to produce stress proteins. In particular, I told you about the example of the MYC proteins that can bind to NK cell receptors and activate NK cells. Well, sometimes over time, tumor cells can just stop making the MYC proteins. They might pick up a mutation where they don't make MYC proteins. If a tumor cell has a variant that doesn't make a MYC protein, that is going to have a selection advantage. It's going to be more able to survive and reproduce and avoid the immune system. And so sometimes we'll see loss of things like the MYC expression that we see. So here you can see loss of MHC again, shown in the middle. So again, we're taking our tumor cell and we're losing MHC. We can also see a situation where perhaps that tumor cell will stop making whatever the tumor antigen is, or just start making less of the antigen. So instead of making less MHC, you make less of the antigen. In some cases, that's possible. So then again, if you have a variant of the tumor that doesn't make the antigen, it's going to be able to evade immune responses better. Same thing with the loss of MHC. Some tumor cells actually start making things that can inhibit T cells. And so some tumor cells actually, because they're just randomly mutating, they might randomly turn on whatever gene. And sometimes they, hit the lottery and turn on something like PD-1 or some other thing that will say, no, T cell, don't kill me. Turn off T cell. And so they will actively inhibit T cells and be able to turn off a T cell that tries to come near them. In fact, we see all sorts of tumors making all sorts of things like Treg cytokines. So lots of tumors make their own IL-10. And so the tumor can kind of act like a Treg and turn off any other T cell that comes near to try to kill it, or lots of other immunosuppressive molecules. So tumors are awesome at making immunosuppressive cytokines and other immunosuppressive molecules. So if T cells come near, the tumor cell turns them off. Some tumors will actually make changes to the extracellular matrix and actually put a wall between them and the T cells. And so sometimes we will actually see a physical barrier get erected because the tumor cell figures out how to make too much extracellular matrix and actually just keeps those T cells away. And then one additional piece of this is that the tumor cell antigen, as we kind of said, is an antigen that's somewhat of a self-antigen, and you probably have that antigen for 20 years, for many, many years. Maybe when you first get that mutation, the T cells are able to respond, but over time, that antigen is going to persist. That antigen is in your body for a long period of time. And so this also, some of this is actually not like actively trying to evade the immune system, but some of it is that we seem to see that those T cells just get exhausted after trying to deal with the antigen for long periods of time. In the same way that we see T cells get exhausted when we deal with chronic viral infection or any other kind of long-term thing. And so the rest of these slides that I'm not really going to have time for we're thinking about ways that we can sort of use our knowledge of the immune system to combat cancer. One of the things we can do is, if it's a virus that is causing the cancer, we vaccinate against the virus. Yay, HPV vaccine. The other thing that I will just say is that in general, one of the problems with a bunch of cancer therapies, and this is sort of true of surgery as we kind of talked about, is that you might not get all the cells, or how do you do the surgery without destroying anything around that area? You don't want to destroy other tissues. Similarly, we tend to do radiation and chemo to kill tumors most of the time. Radiation and chemo is not very specific. It's going to kill any rapidly dividing cell. In fact, you're going to end up, probably needing a bone marrow transplant because we've killed all your immune cells. You're going to kill cells in hair follicles. You're going to kill cells in the GI tract. That the things that we have are not very specific. And so the other place where sort of cancer immunology has really moved forward a lot is we've realized, wait a minute, we have this whole system that's specific for stuff, and we know that we can deal with it can deal with tumors. And so can we use other parts of our immunology knowledge to design either molecules or cells that can influence immune responses, to either wake your immune system back up, or to push your immune system to work, or to directly target the tumor cells. And so the reason why cancer immunology is in fact so big is that we are spending a lot of time now learning ways that we can use this system, whose whole point is being specific, to specifically target tumors. But of course, the key piece to all of this is, do we know what the antigen is? Can we find a unique antigen on the tumor? And can we deal with things like that? So there are lots of ways immunologists are dealing with this. I am out of time, so I'm not going to tell you about them right now. And I will see you tomorrow in lab.